you guys hear me in the back? Yeah, I'll just use this, I think. I'm always afraid of these things because I have friends that have put those on and then wandered off to the bathroom. Or the stage and just died. So let me quickly give you a preface as to sort of the theme for these three talks. And they're very brief talks. And really the goal of these three brief talks is to simply give you guys some sense of the kinds of baseline studies and some of the monitoring studies that are being conducted in the Central Coast Marine Protected Areas. When Caitlin finished her presentation, somebody asked, well, are these things being well enforced? A similar question is, to what extent are we actually monitoring and evaluating the effects of creating these marine protected areas, which was also identified as a fundamental goal in the Marine Life Protection Act initiative. And so we're going to give you and so it's important, I think, for those of you that are interfacing with the public at each of the different state parks to be able to convey some sense of the kinds of monitoring studies that in fact are being conducted in the Central Coast. I'm a kelp forest ecologist. I focus my research interests largely in studying kelp forest ecosystems and the fishes that live in those ecosystems. So when it came time to spin up the monitoring for the Central Coast Marine Protected Areas, I was asked to sort of oversee an effort to monitor the kelp forest ecosystem.
Instructions on where to turn that volume down a little bit.
So very quickly, I want to first give you a feel for where we are conducting monitoring studies in kelp forest ecosystems in the Central Coast. And here's a graph, a map of those study sites superimposed with the marine protected areas. I first want to convey that there's sort of two different kinds of surveys that have been conducted in these marine protected areas. The first is referred to as a baseline survey, the purpose of which was to characterize the state of these populations and ecosystems inside and outside of the protected area across the Central Coast study region. That baseline was funded for 2 years at the initiation of the establishment of the MPAs. In some cases, we were also able to continue those surveys for several years after which, and in fact, the kelp forest surveys that I do were continued on in a monitoring program to help inform eventual evaluations of the protected areas. So you see up here that the baseline was initially conducted in 2007 and 2008. And we conducted these surveys in 14 of the, I think there's either 16 or 17 protected areas across the Central Coast. If you look at the distribution of the protected areas, and I've kind of identified the names of some of those, Terrace Point where we are, right? Terrace Point is the point that we're at right now. Point Lobos, Point Sur, Big Creek, Piedras Blancas, a couple of MPAs down in the Cambria region, and then one at Point Bouchon. You'll notice, where did I do with my little? You'll notice that there is a protected area at Piedras Blancas where we're not doing any surveys. There's also 2 protected areas to the north of this map, one at Año Nuevo and at Greyhound Rock. And we're not conducting surveys at those sites. Can anybody tell me why you think we don't scuba dive at those sites? Thank you very much. I'll pass that on to the technicians so that they don't feel, like a bunch of little wimps and scaredy cats. And so there are some sites that are just too dangerous for us to conduct the scuba surveys. And that's actually one reason why the different other methods that are being used, the collaborative fisheries efforts, submersibles and ROVs, will actually complement the surveys that we do in those areas that we can't. But we did those baseline surveys at 14 of the protected areas. And in the protected areas, I'll show you in the next couple of slides, we pick areas that we survey inside and outside, and they're referred to as cells, where we do a bunch of transects, counting organisms. And so that consisted of 74 cells in this broad barrage of surveys up and down the coast. Then we continued that monitoring past 2008 through to this day, and we have another year of funding the survey again next summer as well. And those surveys were again conducted across those same MPAs in a fewer number of cells up and down the coast. And I'll also just mention that there is, in fact, another protected area further south from this map at Vandenberg, right off Point Conception as well. So I want to give you an idea of how we sort of approach this. And what we do is pick for a given marine protected area. This is the one that's referred to as the Pacific Grove Marine Gardens, which you guys are probably familiar with, between Point Pinos and the Monterey Bay Aquarium. And in this marine protected area, we identify a bunch of potential locations. These are the kelp forest canopies in this very light color that's mapped. And then from those potential sites, we pick at random 4 cells. These 2 cells were sites that we had long-term monitoring studies, so we included those. And then we pick randomly a couple of other cells. And then in each of those cells, we survey a series of transects, which are each of these little purple guys, counting fish. And I'll show you how we do that in a second. And then at a fewer number of sites or locations, we also survey organisms on the bottom, the algae and the invertebrates. And the idea being that by distributing these surveys from the inshore side of the kelp forest to the offshore side of the kelp forest, we survey representatively throughout the kelp forest ecosystem. And what do we sample? Well, because these are conservation with some fisheries related objectives, we're very interested in surveying various population traits. That includes, for example, the density, which is the number of individuals per reef area, and a measure of abundance, or the percent cover for things like algae, that you count individuals. Instead, you just estimate the percentage of the reef that's covered. And then the sizes of those organisms, especially the fishes, because we want to see how the size of fishes change in response to establishing the protected areas, because that is the basis for this increased larval production that Ricky was describing as an objective as well. And then we also combine all those different measures of various populations, species populations, into descriptions of the state of the community, the relative abundance of the various species, and especially those species that have particularly important functional roles in those systems, like the algae that fuels the primary production, and the sea urchins that feed on that and then become prey for sea otters, as well as the sea otters themselves that control the abundance of those urchins. And the way we do that is we swim around and count stuff. And we do that along those transects that I mentioned, and usually there's a diver along the bottom who practically crawls along the bottom, and in a 2-meter wide by 30-meter long strip of the reef surface, they count the number of algae and in particular invertebrates, as well as the percent coverage of those organisms. And of course, we focus on those species that we think are likely most to respond. And those are the ones that are fish. So abalone, sea urchins, those species that we know are being protected from the fishing effort. And then another individual swims above this guy on the bottom and counts fish in the water column, again in a prescribed area. And from that allows us to generate a number of individuals per unit area, which then we can extrapolate over the reef to estimate the abundance of species in the kelp forest. And then we summarize this information, especially for the baseline, by describing the state of these communities, these kelp forest ecosystems at these various locations. And there's some booklets in the back table here that we've generated to summarize that information that I hope you guys will have a chance to look at. So we've done these cool things where we translate the density of the algae and the percent cover into these graphics. They're kind of hard to see here, but if you look at the booklet, the number of little kelp plants, of giant kelp plants, actually scales with the true density of the number of plants that we surveyed at these sites. And then we also look at the surface of the reef and how what the rocky relief is like. That's also summarized down here to describe sort of the geomorphology of those reefs and how similar they are inside the protected areas and outside. And then summarized here are for a bunch of different fish species, their densities given by those horizontal bars, both, again, in this case, at the Point Lobos State Marine Reserve and the Point Lobos reference areas that we've chosen. And so that way we can get an indication or feel for what the state of the fish populations are inside and outside of the protected areas at the initiation of having created these protected areas, which then we can follow through time. And then likewise, similar information for the invertebrates as well. And then I'll finally close just quickly with a comment. I mentioned that we're also interested in how the sizes of individuals in these populations may change. So we actually train people to estimate the length of each fish that they see on their transects. And it's amazing how accurate you can be at this if you're well trained at doing that. And then we bin those size estimates, and that's what's illustrated here. This is what we will refer to as the size structure or the size frequency distribution. And it's simply the percentage of the individuals of a given size or length, in this case, of fish. So this is sort of the size distribution of the kelp greenling, the kelp rockfish, the blue rockfish, inside the protected area, and then similarly in the reference areas outside. And then this is the cumulative number across all of the state reserves and conservation areas, for, in this case, for lingcod, both again inside versus outside. And then we can watch to see whether those sizes eventually change through time, i.e. get bigger in the protected areas, and compare that with the sizes outside to see if in fact we're protecting more bigger fish within the protected areas. That's it for me.